Great, guys. One second, huh? Yeah. So I'm ending the poll now. I'm assuming that I'm clearly audible and my screen is clearly visible to everyone, right? Let me start recording and then we can start the class. Okay. Welcome back to everyone. Welcome back to a new session. Welcome back to a new day, new class, and a new topics. Okay. So yeah, I hope everyone is fine and doing good, right, guys? Great. If you have any doubts, guys, like previous class का कोई doubts है, कहीं से भी कुछ doubts है, please ask. Good evening, Pavana. Good evening. Please ask. Whatever doubt you are having, please ask. Don't hesitate to ask. Okay. Any kind of doubt, I'm open to you people. It's the stage is yours only. Okay. Ask me whatever doubt you are having. Yuvraj, Vidya, Deepika, Gyanansri, Trishala, Tanuja, Soumya, Sinchana, Shreya, Sivraj, Neha, Nandis, Monisha, Monish, Molya, yeah, Likita, Jayant, Jayasuthan, Harsha. Good evening, everyone. Good evening, guys. Okay, welcome back to a new class. Yes. About bioreactors, don't worry. In today's lesson, we'll be talking about that one. Okay. So, before we should start the lesson today, we must revise what we have studied in the previous class. Okay. In the previous class, I taught you about many things like PCR के पहले electrophoresis and in electrophoresis gel electrophoresis के अंदर I told you about density gradient separation as well as isoelectric separation. This isoelectric separation is very much important. Isoelectric point is very much important. Isoelectric point is the point where a strand or a fragment of DNA loses its charge. At that time, the pH value and in pH scale of gel electrophoresis process is called as, that pH value is called as your isoelectric point, right? So isoelectric point के बारे में मैंने बताया था, and after isoelectric point, I told you about competency of host. How can we make the host competent? How can we transfer the plasmid into the host body? And after the transfer of the plasmid into the host body, how the host is now undergoing transcription and translation, and after translation process, how it is producing foreign protein particle. Okay, in today's class we will be talking about foreign protein particle production. Along with that, what is the instrument that is used in this biotechnology process for the production of foreign proteins or foreign products. Okay, and along with that instrument is called as your bioreactors. Bioreactors again are of two types, stir type and sparger type. We'll be talking about these two things separately. After bioreactor, we'll directly go to the next part. The product that we are getting through bioreactor, in bioreactor, that product is not completely pure. जो product हम लोगों को bioreactor में मिलता है, that product is not completely pure. Okay, guys. So what we need to do, we need to isolate that product. We need to extract that product. After extraction of the product, we need to purify the product. After purification, packaging is required, and after packaging, we have to store it, or else we can commercialize it also. Okay, so all these process, extraction से लेकर के commercialization तक, everything, all the process included in a single step only, and that single step is called as downstream processing, right? So in today's lesson we'll be talking about all these things in detail. सही है. उसके बाद if you are getting sometimes, then we'll directly move to the next biotechnology chapter. Biotechnology application in agriculture, application in medicine, and application in other genetically modified organisms, and all other things. ठीक है, medicines, pharmaceutics, किसमें biotechnology का application use कैसा है. We'll be talking about all those things in detail if we get time. My screen is showing YouTube live. Okay. Is it visible now, guys? Is it visible now? Okay. Thank you, Sivraj. Thank you, Amit. Now clear. Thank you. Okay, so let us start the topic. आज का लेसन स्टार्ट करने से पहले here is a question for you people. Question is like, when isolating a pure DNA from bacterial cell, the cell should not be treated with which of the following? Okay, option A lysozyme, option B proteases, option C ribonuclease, option D deoxyribonuclease. I am launching poll for this question. You can answer in the poll. I hope this question is very much easy and you can answer it in the poll as well as in the chat box. Wow, that's a great thing. I got many answers here. Okay, correct Soumya, correct Srinidhi. Yes, correct Nandis, correct Kishore, correct Amit. Everyone is correct. Very good guys. Correct Harsha. Exactly. What about rest of the people? Exactly. D. Exactly, Nandis. Your answer is correct. Okay, but you have to choose the correct option from this all these options. A, B, C, D में से किसी एक को treat करना है. Okay, किसी एक को choose करना है. Okay, correct Nandis. Exactly. Correct Disha. Correct Dikshit. Correct Swati. Okay, last 15 seconds remaining. 41% people have chosen their answer so far. I want the rest of the people to answer this question. Guys, please answer. Please answer it. Do it first. Correct, Swati. Very good, guys. I'm ending the poll now, and I'll be sharing the result with you people. Before sharing the result, 1 second. Let me mute everyone. Okay. So now, okay, Nandis, I'll be asking you tougher question in the next class only. Okay. ठीक है, अभी सही है ना? चलो, 1 second, guys. Just give me 1 minute. Let me make Manjula co-host. Okay. Manjula, you are the co-host now. Okay. Observe the class carefully. Okay, guys. So the result is like 12% people have chosen option A, 12% people have chosen option B, 4% people have chosen option C, and 72% people have chosen option D. Let me stop sharing, then I'll explain you. We want to extract DNA from bacterial cell. What we want to extract? We want to extract DNA from bacterial cell. Okay. If you want to extract DNA and if you are applying deoxyribose nuclease or DNase enzyme, DNase enzyme अगर apply करते हो, it means it will completely digest the DNA. Will not be able to extract DNA. This DNase enzyme will be completely digesting the DNA. So this will be the correct answer. आराम से cleanly आराम से answer कर सकते हो. Very simple question. Lysozyme to digest the cell wall. Lysozyme to digest the cell wall. Proteases to digest the plasma membrane. Ribonucleases to digest the RNA. ठीक है, प्रोटीएजेस टू डाइजेस्ट प्लाज्मा मेम्ब्रेन एज वेल एज प्रोटीन को भी डाइजेस्ट करने के लिए यूज होता है. ओके, सो दीस थ्री थिंग्स कैन बी डाइजेस्टेड बट वी डोंट हैव टू डाइजेस्ट DNA because we want to extract DNA from the bacterial cell. Is that clear to everyone? So option B will be the correct answer. That is deoxyribonuclease should not be given to the bacterial cell in case if you want to extract DNA from it. Is that clear to everyone? Okay. Okay, उषा का एक छोटा सा क्वेरी है, मैं बता देता हूँ. उषा, मंजुला is your madam. Okay, she is biology madam. That's why I made her co-host. Amit, I'll be doing that question solving and everything. I'll be doing one class. ठीक है, that will be on Friday. Question solving and revision will be on Friday for this particular chapter. Don't worry about this one. ठीक है? ओके, नो प्रॉब्लम. चलो फिर आज का कॉन्सेप्ट स्टार्ट करते हैं. आज का टॉपिक है ऑब्टेनिंग फॉरेन जीन प्रोडक्ट. ऑब्टेनिंग फॉरेन जीन प्रोडक्ट इन द सेंस व्हाट आई डिड, आई अप्लाइड और आई inserted the recombinant DNA into the host and now the host will be undergoing transcription and translation. Inside the host, transcription will happen. Okay, and after transcription, mRNA will be produced inside the host body. And from mRNA, this mRNA will undergo translation, and after translation, protein will be synthesized. The protein which is synthesized here is a foreign protein because it is produced from the foreign DNA. That's why it is called as your foreign protein or recombinant protein. So recombinant protein will be synthesized. Recombinant protein will be synthesized in this way. Transcription translation के बाद recombinant protein will be synthesized. Is that clear to everyone, guys? If it is clear to all of you, click on yes option. जल्दी से respond करो. Yes, exactly. आनंद इसको vote करेंगे, ठीक है? Yes, yes, yes. Click on yes option if it is clear to all of you. This very simple concept I just explained you. 2 minutes का एक छोटा सा concept था. I explained you. लिखित मंजुला, sorry, not मंजुला. लिखित, किशोर, दीपिका, ज्ञानश्री, चेतना, अनन्या, अमित, Swati, click on yes option if it is clear to all of you. Bhagyasri, click on yes option if it is clear to all of you. Okay. Okay, we can move to the next slide now. This can be obtained अगर single individually एक host को लेकर के, if you are doing the same role like transcription, translation, and then protein extraction. Okay, if you are doing that by taking a single host, then it will be like taking much time. It will take much time. That's why what we do, we put this host in a medium and in a container, medium containing in a container, contain in a container. That particular container is called as your bioreactor. In the next slide only I'll explain you. We have to take some bioreactors. 2 types of bioreactors are there. Inside these bioreactors, we put our raw materials. क्या put करेंगे? We will put raw materials. We'll put raw materials. यहाँ पे हम लोग raw materials डालेंगे. What are the raw materials here? Raw materials क्या है? Raw materials are like host containing gene of interest. Host containing gene of interest. Okay. Second thing, the culture medium. Culture medium in the sense, in the medium we have to provide some nutrition. Culture medium के अंदर nutrition वगैरह डालेंगे जहाँ पे host can survive. Okay. Along with that, एरिएशन मस्ट बी happening. एयर प्रोवाइड करेंगे, एरिएशन होना चाहिए, एयर प्रोवाइड करेंगे, ठीक है. These 3 things must be provided in the bioreactor. Bioreactors are the instruments. Bioreactors are the instrument. ठीक है. Okay, Usha, don't worry. ठीक है. So bioreactors are of 2 types. These are the instruments. These are the instruments which are used in biotechnology process. Bioreactors are of 2 types. First one is your stir type and second one is your sparger type. This first one is your stir type of bioreactor and second one is your sparger type of bioreactor. Sparger type of bioreactor. Is this clear to everyone? When you look into the stir type of bioreactor, these 2 bioreactors are a little bit different. I'll explain you how they are different. Okay. In stir type of bioreactor, a motor will be present on the top side, in the top side. Motor रहेगा. To the motor, a shaft is generally attached, like a rod attached होता है, ऐसे नीचे की तरफ जाता है, red color का rod. I can show you. This is the rod. Motor रहेगा और motor के साथ attached होगा एक rod. Okay. To this rod, many shafts will be attached. These blades are called your shafts. ये blades ऐसे attached रहेंगे और blades और shafts I can say. What will happen? Whenever I am turning on the motor, this motor will rotate the rod, and along with the rod, this particular blade or shaft will rotate in this direction. अभी क्या होगा, shaft और blade जो है ना, ये rotate करेगा, ऐसे circular motion में rotate करेगा. ठीक है. Whenever I am turning on the motor, the shaft or the blades are moving like this. It is rotating like this. Okay. This rotation of the blade or shaft will help in mixing of the raw material. जो भी raw material हम यहाँ पे डाले हैं, उसका mixing में ये help करेगा. It will be helping in the mixing of the raw material. Raw material will be provided in the bioreactor only. Beside this shaft and motor, these bioreactors are having many other sites. What are the sites? Like there will be an acid-base pH control port and one steam sterilization control. This will be like lowering the temperature and increasing the temperature. Temperature increasing and temperature lowering function will be performed by the steam sterilization. Steam for sterilization. Okay, this particular port. This port will be adjusting the pH It will be increasing the pH whenever required, and it will be decreasing the pH whenever required. pH को increase or decrease करेगा. Then how can we get to know that the pH is increased or the pH, what is the pH level or what is the temperature level? For that, in this stir type of bioreactor, here in this side only, one thermometer will be there and one pH scale will be there. ये रेड कलर का मैंने जो बनाया, रेड कलर का ये अपना pH स्केल है, pH स्केल बनेगा ये, जो कि मेजर करेगा pH को. एंड ये ब्लैक कलर का जो मैंने बनाया, दिस विल बी योर थर्मोमीटर. Is that clear to everyone? This will be measuring the temperature, and pH scale will be measuring the pH of the medium. Okay, in this way all the raw material will be like given into this bioreactor. Inside the bioreactor, motor will be turned on. This motor will, like when the motor is turned on, blades and shafts are there rotating like this in a circular motion. Blades and shafts is rotating in a circular motion. Whenever this is rotating, this will help in aeration as well as mixing of the raw material. Whenever the mixing is happening, okay, it will form some foam on the top side. So top side में क्या होगा, थोड़ा सा foam बनेगा, FEOM, foam बनेगा और broth बनेगा, foam और broth बनेगा. This foam or broth is broken down by the first shaft. यहाँ पे जो first shaft है, ये foam breaker है. First shaft is also called as foam breaker. Foam breaker, ये first shaft इस foam को break करेगा, ठीक है. And foam break करने से aeration अच्छे से होगा. Foam break करने से aeration अच्छे से होगा. Is that clear to everyone? Right? But whenever you are filling this bioreactor, you must have to fill this bioreactor up to 3/4 part of the bioreactor needs to be filled. 1/4 of the bioreactor needs to be every time 3/4 portion जो होगा वो filled होगा bioreactor का. Rest of the 1/4 portion it should be empty. 3/4 portion जो होगा ये filled होना चाहिए. And rest of the 1/4 portion ये empty होना चाहिए. This must be empty. ठीक है. For the proper aeration के लिए ये होना चाहिए. Is that clear to everyone? How? Now I got your doubt. Now this you have raised your hand. Okay, you have raised your hand. I got it. I'll check whatever doubt you are having. Action stick, mixing grinder, pH scale, foam. Can't we fill it one? No, not exactly. We cannot fill it 1, 2. We have to fill it 1, 3/4. Huh? We cannot fill it 4 out of 4. We can fill it 3 out of 4, 2 out of 4, or 1 out of 4. No problem. But we cannot fill it more than third part of the out of 4th. 4 part में से third part fill कर सकते हैं, third तक fill कर सकते हैं, 3/4 fill कर सकते हैं, but not more than 3/4. We can fill it less than 3/4, no problem. Less than 3/4 इसको fill कर सकते हैं, no problem. ठीक है. Foam क्या होता है? Foam are like broth और foam होते हैं. Foam means like what I can say. Do you use soap? Soap use करते हैं ना तुम? तो soap जब use करते हो तो soap अपने body में लगाने से at that time foam are produced. Foam produce होता है. That foam will be produced here, and that foam will be broken by this foam breaker or first shaft in this stir type of. Yes, exactly, serving foam we can say. Okay, and in sparger type what happens? Sparger type is slightly different from the stir type. In stir type motor were present, but in sparger type no motor will be there. No motor will be there. कोई motor नहीं होगा. In sparger type, on the lower parts of the sparger type there will be a structure. This particular structure is called as your sparger. ये sparger की तरह होगा, इसको sparger कहा जाएगा, right? On this sparger many pores will be there. Minute pores will be there. छोटा छोटा pores होगा यहाँ पे. You can see here. Minute pores will be there. Through these pores, air will be released into the tanker. Air will be released into the tanker. Air will be released into the tanker. What is released here? Air is released into the tanker through this sparger. इस sparger के जो spore है, spore के मदद से छोटे like air molecule release किया जाएगा. This release of air molecule from the bottom side, कहाँ से release हो रहा है? This is released from the bottom side. This will help in mixing of the material. Whenever it is releasing from the bottom side, air is released from the bottom side. Suppose this is your container. 1 second, Nandis, I will come to your doubt. 1 second. Okay. I got it. You have raised your hand. Suppose this is your like container. ये container है. नीचे से air आएगा. From the lower side only air will come. Air आएगा. This air, whenever this air is coming from the bottom side, this will create some pressure and it will help in the mixing of the raw material. And this is how Sparger type function. Only difference between Sparger type and motor type is in motor type or in stir type, there will be a motor which will be regulating the function of mixing. But in Sparger type, Sparger will be there, which will release the air from the bottom side, and this released air will help in mixing and aeration of the raw material. This will be the only difference between Sparger and stir type. Okay. Is that clear to everyone? समझ आया बात? Okay, the question is like अभी आया है नंदिस का एक छोटा सा doubt. The doubt is like how this steam sterilizer will be sterilizing the steam. See, what happened? Suppose this is your bioreactor. I am making it outside only. Okay, for your clarification, this is suppose your bioreactor. This is suppose your bioreactor. In this bioreactor, there will be an outer covering of the bioreactor. Outer covering means there will be a jacket of this bioreactor. Bioreactor का एक छोटा सा jacket होगा जो कि इसको बाहर से completely cover करेगा. It will cover it from the outside. It will cover it from the outside. इस तरीके से. It will cover like a jacket. It will cover like a jacket. Is that clear to everyone? Just wait guys. It will cover like a jacket. बाहर से इसको completely ऐसे cover करेगा. इसको completely it will cover like a jacket from the outer side. In this jacket, what you can see on this side, there will be an opening, and on this side there will be an opening. What will happen now? Through this opening, we have to provide water, and through this opening, the water will go out. This is a jacket. This jacket, like this is present out of this particular tanker. Bioreactor tanker की ये बाहर होगा. Okay. Now what happens? If the temperature is low, at that situation, steam or hot water will be provided. Hot water will be provided through this jacket. Hot water will get into this jacket, and this will accumulate all around. इसके चारों तरफ accumulate करेगा, and this will help in increase the temperature. This will help in increase the temperature inside the container or inside the bioreactor. And after the increase of the temperature, again this water will get out through इधर से ये बाहर निकल जाएगा water. This is how steam sterilizer work. Is this clear to everyone? अभी आया समझ में नंदीस? ठीक है, पाउच की तरह काम करेगा. Exactly. Is this clear to everyone? Sterilization means not exactly making it pure. Sterilization means इसको constant करना, normal करना. It means like make it normal. Sterilize means whenever the temperature is going high, you have to decrease down the temperature, and whenever the temperature is going low, you have to increase the temperature. That is your sterilization. Not exactly like making it pure or making it completely sterilized. Okay, sterilization means you have to make a proper condition. You have to create a suitable condition. That is your sterilization. Is that clear to you? स्पार्जर टाइप का रिपीट करने के लिए नंदिस का ये है. सो लेट मी रिपीट दिस स्पार्जर टाइप. इन स्पार्जर टाइप मोटर टाइप के जैसा था, स्पार्जर टाइप में भी वैसा ही है. नो डिफरेंस बिटवीन मोटर टाइप एंड स्पार्जर टाइप और स्टीर टाइप. ओनली वन डिफरेंस इज देयर. इन स्टीर टाइप motor was present, but in case of sparger type, no motor will be there. The role of motor was to mix up all the materials, raw material को mix up करने के लिए help करना था, right? But in sparger type, on the bottom of this sparger type, there will be a sparger, and in the sparger, minute pores will be there, through which air will be released into the container, from the bottom side. When the air is released from the bottom side into the container, this air will help in mixing of the raw material and all other material. Mixing में help करेगा, aeration में help करेगा. Okay, and this is how product will be produced, and the product will be released from the bottom side only. यहाँ पे छोटा सा opening होगा. This will be the product side. Product hole. यहाँ से product निकलेगा. Is this clear to everyone? अभी समझ आया सबको? हाँ मोनिसा, next process is your protein like purification का जो बात है ना, that will be we will be doing that one. Amit, the best one is your stir type, क्योंकि मोटर है यहाँ पे, and air will be mixing it, but air का जो mixing होगा उतना ज्यादा like properly mixing नहीं होगा. Okay, that stir type is like is the best one. Stir type is the best one. Nandis ka doubt is sparger means what? Sparger means it's like a badminton type structure. It's like this. It is a structure like this. I can show you. This is your sparger. रैकेट, बैडमिंटन रैकेट जो होता है ना उस टाइप का होता है. यहाँ पे छोटे छोटे पोर्स होते हैं. These are minute pores. This is your sparger. Sparger is a structure. Is this clear to you, Nandis? अभी अभी आया समझ में, right? The name is just because of that one. Like इसका structure is sparger type होता है. Go and search in the Google, you will get it. Okay, no problem. ठीक है, समझ आया? Molya, the bioreactor are only used for mixing raw material. Not exactly, Molya. Bioreactors are used for mixing the raw material. Raw material and host, they will be mixed inside the bioreactor, and inside the bioreactor only, after the proper mixing and proper aeration, this host will undergo transcription and translation, and the product will be produced. Okay. Okay, Nandis, I will not be taking your name again and again because you are a good student. That's why I am taking your name again and again. Okay. Well, in bioreactor, what happens? Host will be producing the a protein will be produced inside the bioreactor, along with all the mixing will be happening inside the bioreactor. Okay. So bioreactors are generally used for the production of the things or proteins or the needy substance. Okay, by providing optimum temperature, proper amount of food particle, proper amount of nutrition, culture medium, and everything will provide, and raw materials. And after that, we'll get up to 100 to 1000 liter of culture in a very few or very less time period. Okay, this is all about your bioreactor. From the bioreactor, we'll be getting the product. From the bioreactor, we'll be getting the product, and that product is your foreign gene product, like foreign proteins or anything. We'll be getting the products as proteins. Proteins के हिसाब से हमको मिलेगा. These proteins are not pure proteins. These are impure products. These products are impure products. This product needs to be pure. This product needs to be purified. This product needs to be purified. For the purification of these products or these proteins, we have to go through another process. That process is called as your downstream processing. Right, guys? We'll be talking about that downstream processing. Before that, again, I got few doubts, I guess. Yes, exactly. Nandis, mixing होता है इसीलिए होता है. Monisha, inside the bioreactor, host will be present. Host will be present. Okay, Monisha का question है, how protein are produced inside the bioreactor? Host will be provided or kept in the bioreactor with the proper amount of culture medium and nutrition and everything. Now this host is aerated properly and nutrition is provided to this host properly. This host contain our gene of interest. Inside the host body, it will undergo transcription. जो भी gene of interest होगा, it will undergo transcription, and after transcription, mRNA will be produced, and after mRNA, translation will happen, and translation के बाद protein will be produced. In this way, protein will be produced. Is this clear to you now? ठीक है, central dogma will happen exactly. समझ आया मोनिशा? ठीक है, in this way protein will be produced. ठीक है. Before going to the downstream processing wala part or what I can say like purification of protein wala part, here is a question for you people. Question is like, elution means what? Elution means what? Okay, option A, separating the restricted DNA fragments on agarose cell. Option B, staining the separated fragment with ethidium bromide. Option C, cutting out of the separated band of the DNA from the agarose gel and extracting them from the gel piece. Okay, and option D is construction of rDNA by joining the purified DNA fragments to the cloning vector. Which of the following is correct? I'm launching poll for this particular question. You can answer in the poll. That's a great thing. Like I got answers from 20 people before the launching of the poll. Only 20 answers आया. Oh, एक ही पर्सन का आंसर है इतना सारा. Correct. Molya, correct. Not exactly Diksha and Likhit. Think about this one. Correct, Nandis. Correct, Sinkana. Not exactly Swati. Correct, Diksha. Correct, Nandis. Exactly, Nandis. Very good. Correct. No, Likhit. No, not exactly. Think about this one once again, Likhit. और एक बार सोचो, then you can answer properly. You can answer correctly. Yeah. Think about this one once again. You can do it. Last 25 seconds remaining, 35 seconds gone, and 27% people have chosen their answer so far. I want rest of the people to answer this question. Guys, do it first. I want everyone or rest of the people to answer this question. Do it first, guys. Jaldi se kar do. Yes. Do it fast. Correct, Tanuja. I'm ending the poll now. Sharing the result with you people, and the result is like 13% people have chosen option A, 26% people have chosen option B, 61% people have chosen option C, and no one have chosen option D. Let me stop sharing, then I'll explain you. Illusion is the separation. It is the separation or isolation. One second, Nandis, you have raised your hand once again. I'll be coming to. Okay, you are correct, Nandis. Separation of DNA from agarose gel plate. Separation of DNA from where? Agarose gel plate or agarose gel. Agarose gel से DNA को separate करने का जो भी चीज होता है, that separation of DNA from agarose gel is called as elution, and this is performed by a technique. This elution is performed by a technique, and that technique is called as your blotting technique. Is that clear to everyone? That technique is called as your blotting technique. What I explained you yesterday only, last class में, जो कि मैंने तुम लोगों को बताया था, blotting technique के बारे में बताया था. And out of all the type of blotting technique, for the separation of the DNA from the agarose gel plate, or to perform elution process, we need southern blotting. Which blotting technique we need? We need southern blotting technique. Southern blotting technique. Is that clear to everyone? Okay. So option C will be the correct one. That is cutting out of the separated band of the DNA from agarose gel. Agarose gel से DNA को निकालने का technique. That process is called as your elution. This process is performed by a technique, and that technique is called as your blotting technique. Is that clear to everyone? Okay. Separating restricted DNA fragments on agarose gel, that is your gel electrophoresis. First option is your gel electrophoresis. This is your gel electrophoresis process. Gel electrophoresis process. Okay. Right. This is not elution. Staining the separated DNA fragments with ethidium bromide. This is like this is staining only. This is staining only. This is not your elution process. Right. Ethidium bromide के साथ staining किया जाता है, so that it can be visualized under X-ray or any other material. X-ray में ये visualize हो जाए करके. Next one is your construction of rDNA by joining the purified DNA fragments to the cloning vector. That is your rDNA का formation, and I can say this one is your ligase के वजह से ये होता है. ठीक है. So this is also not your answer. Option C will be the correct answer. I hope this is clear to everyone. हाँ, correct, Sivraj, exactly, Sivraj. Okay, let's move the last part, that is the downstream processing. And I told you in the downstream processing what happens. The product is produced now. The product or the protein is produced now. This protein will be isolated. First step, this protein will be isolated at first or separated from the tanker. After the separation or isolation of the protein, this protein will undergo purification. This protein will undergo purification. This will undergo purification. Is that clear to everyone? Purification हो जाएगा. And after purification, one second guys, this will undergo the next process, and that process is, let me take blacking here. Okay, so this will undergo packaging. Purification के बाद packaging होगा. Packaging के बाद this can be stored, or I can say preserve. Preserve इसको कर सकते हैं या फिर store कर सकते हैं, and then we can commercialize it. We can commercialize, or we can release it in the market. Commercialize it, or it can release it in the market. Okay, so downstream processing includes all the process after the production of the protein. to test the efficiency, safety, recovery, cost, and everything, quality control, everything will be performed inside this downstream processing. Downstream processing के अंदर everything हो जाएगा, quality control check किया जाएगा, safety इसका कितना है, efficiency कितना है, recovery कितना है, cost efficiency क्या है, everything will be checked in this process. Okay, isolated, purified, and packaging and preserved, and after that it will get commercialized. This will include your downstream processing process. Okay, this will be the last step, and here your chapter is completed. Biotechnology principle and process is completed here only. Is that clear to everyone, guys? Right, now we'll move to the last part. One more question is there here. Okay, question is like, which of the following is not a component of downstream processing? This question has been asked in NEET 2016. Now here only I explained you the processes or steps of the downstream processing. Okay, options are option A expression, option B preservation, option C purification, option D separation. I'm launching poll for this particular question. You can answer in the poll. ठीक है, मैंने पोल लॉन्च कर दिया है क्वेश्चन में. You can answer. Correct Salini, correct Somya, correct Somya, correct Diksha, exactly correct Nandish, correct Mulya. Yes, 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 correct Anuradha, very good Anuradha, very good. Yes, I want everyone to choose their answer properly and correctly. Correct Sinkana. 30 seconds gone, last 30 seconds remaining. 29% people have chosen their answers so far. I want the rest of the people to answer this question. Okay, Harsa, I'll explain you this one. ठीक है, मैं बता दूंगा, no problem, Harsa. I'll explain. Once again, I'll repeat this downstream processing वाला part. ठीक है. Give one day today example of including all steps of the biotechnology. I'm not getting you on this. One product. One product. We'll be talking about all those things. Don't worry on this. Okay. Whenever you are talking about this, which one, like application वाला part, application में बहुत सारे examples will be talking. Applications में बहुत सारे examples के बारे में will be talking. Okay. In biotechnology, many things are produced. By the help of this biotechnology, medicines are produced. Okay, by the help of this biotechnology, you people might be drinking this one. माजा मिलता है, ठीक है, माजा. फिर आता है, एक आता है अपना मैंगो जूस, बहुत सारे आते हैं ना, मैंगो जूस, लाइस, माजा एंड एवरीथिंग. ओके, दैट आर प्रोड्यूस्ड बाय द बायोटेक्नोलॉजी प्रोसेस ओनली. ठीक है. Yes, Shivraj, this is a very small chapter. Slice होता है, right? Slice होता है. And हाँ, how impure protein is isolated? Okay, I'll explain you. Don't worry, Manisha. ठीक है, fruity, masa, everything will be produced through this process only. Let me end the poll and then I'll share the result. The result is like 79% लोगों ने choose किया option A को. That's a great thing. 4% people have chosen option B, 8% people have chosen option C, and 8% people have chosen option D. Let me stop sharing, then I'll explain. When I am talking about downstream processing, okay, in the bioreactor, on the bottom of the bioreactor, there will be an opening. That opening is for the collection of the product. Products will be collected from here. Products will be collected from here. The synthesized protein will be accumulating inside the medium now. जो भी protein synthesize हुआ उस medium के अंदर अभी accumulate कर रहा होगा. Whatever protein is synthesized by the like host will be accumulating inside the product on in the bioreactor only. We have to take out the product and we have to make the downstream processing of this product. That is your first one is your isolation. We did isolation or separation. Isolation किया हम लोगों ने निकाल दिया. After isolation, we'll have to filter it. We have to filter it, or I can say filter के साथ साथ इसको separation भी बोल सकते हैं. After separation, we have to purify this one. We have to purify this one, and after purification, this needs to be packed. Packed हो जाएगा, and after packaging, it needs to be stored or preserved. If necessary, it needs to be stored or preserved. and if it is not necessary, then it will be like commercialized. These all steps include your bio like downstream processing, included in your downstream processing. And the question is asking about which is not a component of the downstream processing. Expression is not a component of the downstream processing, since preservation is a process, purification is a component, and separation is also a component. Expression is not a component of the downstream processing. Option A will be the correct answer. Option A will be the correct answer. That will be the answer. Okay. Yeah. Correct. Exactly. Is this clear to everyone, guys? So this is all about your this chapter, and उसके बाद हम लोग चलेंगे डायरेक्ट और इसका मॉड्यूल क्वेश्चन सॉल्व करना. आई विल बी शेयरिंग द मॉड्यूल टुडे ओनली इन द ग्रुप. मॉड्यूल सॉल्व करना इसको मैं शेयर कर दूंगा ग्रुप में आज ही. ओके, so let us move to the next topic now. We'll start the next chapter. Before that, just ask me your doubts, guys. If you have any doubt from this particular chapter. CFU का मतलब होता है check for understanding. Okay, so just let me guys, please ask me if you have any doubt from this particular chapter. इस chapter से any kind of doubts you are having, please ask me. Please ask me if you have any kind of doubts in the chat box. Otherwise, I will start the new chapter now only. And if it is clear to all of you, click on yes option. Yuvraj, Meghana, Ruchita, Rajkumar, Likit, Gagandeep, Danush, Yashashwini, Vidya, Viva, Virshini, Varshini, Tanuja, Usha, Srinidhi, Somya. Click on yes option if it is clear to all of you. Shreya, Salini, Puneet. Click on yes option if it is clear to all of you, guys. Jaldi se yes option पे click करो. Okay. ठीक है. I'll be explaining bioreactor once again, no problem. ठीक है. Which is the best function performed in the lab in biotech? Okay. उषा का क्वेश्चन ये है कि like biotech में कौन सा बेस्ट फंक्शन किया जाता है in biotech. Like many functions are there. Even if whatever vaccine you are taking for this COVID-19, that vaccine is also prepared. ठीक है. That vaccine is prepared by the help of this biotechnology only. ठीक है. That vaccines is like prepared in the biotechnology lab only. ठीक है. चलो. If you have any other doubts, please ask. I'll explain. Insulin is also produced by the help of biotechnology process, and that is application of biotechnology के अंदर वो insulin आएगा. I'll be explaining you about that one. Medical science में. In vitro fertilization is also a biotechnology process. Okay. Downstream processing है हर्षा का doubt. Let me explain downstream processing once again. Okay. Please guys stay in the class and with patience. Pallavi, don't go in depth of the this purification. Purification के in depth में मत जाओ अभी. ठीक है. Don't go in depth of the purification. Purification का in depth we'll be talking about this one. What about Nandis, like why you put this message? Is there any issue? Yes. Okay. So बायोरिएक्टर का थोड़ा सा मैं बता देता हूँ. Okay. Bioreactor का देखोगे तो bioreactor are the like two types of bioreactors are there, stir type and sparger type. These bioreactors are the instrument where raw material will be put or a host, culture medium, aeration, everything will be provided in the bioreactor, and it will allow to grow. The hosts are allowed to grow and hosts are allowed to undergo transcription and translation and to produce the protein or other things. Okay. This bioreactor are of two types, stir type and sparger type. In stir type and sparger type bioreactor, in stir type, motor will be present, and to the motor a shaft will be attached, and to the shafts many blades will be there. That blades will rotate when the motor is turning on. When the motor is turned on, the blade will rotate, and this rotation of the blade will help in mixing of the material and aeration of the material. Okay, आया समझ में सबको? ठीक है. And in sparger type, this motor will be absent in sparger type, and in sparger type, sparger like structure will be there on the bottom of this sparger type bioreactor. This sparger type bioreactor has a sparger in the bottom side, and from the bottom only, from the sparger, air will be released into the container or bioreactor. When the air is released from the bottom side into the bioreactor, this released air will help in mixing of the material. मटेरियल का मिक्सिंग में ये हेल्प करेगा. आया समझ में सबको? ठीक है, आई होप दिस इज क्लियर नाउ. ठीक है ना? ओके, ठीक है, चलो लेट्स मूव टू दी नेक्स्ट टॉपिक. नेक्स्ट टॉपिक इन द सेंस योर लास्ट सेकंड वाला चैप्टर आ जाएंगे अपना बायोटेक्नोलॉजी का एप्लीकेशन में. विल मूव टू दी बायोटेक्नोलॉजी का एप्लीकेशन. अभी directly we'll move to the biotechnology application. Yeah, biotechnology and its application. Applications of biotechnology are of many types. Like we can use biotechnology in various fields. In our like constantly again present example अगर लेते हैं हम लोग, in present example biotechnology is used in your which one, like medicine में अभी यूज होता है इसका अच्छे से. इन मेकिंग कोविड-19 वैक्सीन, जो सील्ड कोविशील्ड है या फिर कोविशील्ड है या फिर अपना कोवैक्सीन है, दैट इज प्रिपेयर्ड इन बायोटेक्नोलॉजी लैब ओनली. भरत बायोटेक लैब, और भरत बायोटेक इज द लोकेशन फ्रॉम वेयर इट इज द लैब फ्रॉम वेयर कोवैक्सीन इज प्रिपेयर्ड. ओके, so covid vaccine is prepared from by the help of this biotechnology only. So biotechnology can be used in various purpose के लिए biotechnology का यूज होता है. हाँ मोनिसा, exactly. In case if it is a large plant or animal, in that case we don't have to insert into the bioreactor. Okay, the plasmid is directly inserted in our body and that will start producing your gene of it, like product. जो भी प्रोटीन तुमको चाहिए था वो प्रोडक्ट तुम्हारा, वो प्रोटीन that will be synthesizing in our body only. Okay Manisha, we don't have to be like, we don't need to insert plants and animals into the bioreactor. Only bacteria and all small microbes can be inserted into the bioreactor. We are not inserted in the bioreactor. ठीक है. E. coli can be inserted into the bioreactor. E. coli is a bacteria, Escherichia coli. ठीक है. Okay. Bioreactor are only and only used if you want a product in much amount. If you want a product in much amount, then only bioreactors are used. Is this clear to you? ठीक है. Now, in application of the biotechnology, we'll start talking about many things. But before that, here is a question for you people. Question is like, which of the following restriction sites is located within gene for tetracycline resistance in plasmid PBR322? In plasmid PBR322, okay, many restriction sites are there, and in restriction sites, याद करो यहाँ पे इसी लोकेशन पे tetracycline था, TETR था. Along with TETR, two more restriction sites were there. The question is asking about the restriction site which are present along with the TETR or tetracycline resistance gene. Okay, I'm launching the poll now. Options are like BAM H1, option B PST1, option C CLA1, option D PBU2. I'm launching poll for this particular question and I hope you can answer this question. ठीक है, चलो जल्दी देख लेते क्या क्या आता है रेस्पॉन्स तुम लोगों का. मैंने पोल लॉन्च कर दिया. यू कैन आंसर इन द पोल गाइस, यू कैन आंसर इन द पोल. करेक्ट सौम्या, नॉट एग्जैक्टली. स्वाति करेक्ट. नंदीश करेक्ट. मोल्या करेक्ट. शिवराज करेक्ट. जयंत वेरी गुड. जयंत वेरी गुड एवरीवन. करेक्ट. किशोर एग्जैक्टली. किशोर फ्रॉम जेनवियासन. अनुराधा वेरी गुड अनुराधा, that's correct. 25% people have chosen their answer. I want rest of the people to answer this question. Correct Nandis, you are very absolutely correct and super active in the class. ठीक है, ठीक है, जल्दी करो. Last 15 seconds remaining. I want everyone to answer this question. Do it fast. 33% people have chosen their answer. Not exactly. Diksha correct. Sinchana very good Sinchana. Diksha, think about this one once again. You can do it Diksha. और एक बार सोचो, you can do it. Okay, amending the poll now, sharing the result with you people, and the result is like 64% people have chosen option A, 16% people have chosen option B, 12% people have chosen option C, and 8% people have chosen option D. Let me stop sharing, then I'll explain. ठीक है, ठीक है. So हाँ, correct Chakshutha. Right. So when you talk about TETR, two restriction sites along with tetracycline resistance gene is present in this location. First one is your BAM H1 and second one is your SAL1. Okay, so you can find out like यहाँ पे कहाँ पे SAL1 है और BAM H1 है. First option में BAM H1 है, option A will be the correct answer. PST नहीं, CLA नहीं, PVU नहीं, none of these. Okay, only BAM H1 is the correct answer. That option A will be the correct answer. That is present the region where TET or tetracycline resistance gene is present. Okay, this kind of question are generally asked. Let's move to the next part. That is biotechnology का applications in different part. Biotechnology can be used in biopharmaceuticals. That is pharmaceuticals means जैसे कि हमने वैक्सीन्स के बारे में पढ़ा, for the preparation of vaccine and medicine. वैक्सीन मेडिसिन के लिए इसका यूज होता है, वैक्सीन एंड मेडिसिन के लिए इसका यूज होता है. थेराप्यूटिक्स मेडिसिन के लिए, थेराप्यूटिक्स मेडिसिन के लिए. ओके, diagnostic to detect certain disease like a PCR technique, it is coming under biotechnology, and PCR technique is used to detect certain diseases. कुछ diseases एलाइजा एक टेस्ट होता है जिसके मदद से टेस्ट डिटेक्ट किया जाता है, HIV एड्स को डिटेक्ट किया जाता है एलाइजा टेस्ट की मदद से. जेनेटिकली मॉडिफाइड क्रॉप्स के बारे में पढ़ेंगे, हाउ कैन वी मॉडिफाइड द क्रॉप बाय द हेल्प ऑफ दिस बायोटेक्नोलॉजी, उसके बारे में पढ़ेंगे. प्रोसेस फूड, बायो रेमिडिएशन, waste treatment, energy production, these are some applications of the biotechnology. We'll be talking about each one of these separately. We'll start with the pharmaceutical medicines. Correct Nandis and Sivras. We'll start with the agricultural part. How biotechnology can be used in agriculture. We'll start with that one. Okay, 3 critical research area are there for the biotechnology. Go through this one. I'll share with the with you people this particular slide. इसको देख लेना. Okay, now applications in agriculture, how biotechnology can be used in agriculture. We'll be talking about those things. Okay, agriculture can be of different type, or agriculture is performed in 3 different ways. First one is your agrochemical based agriculture, second one is your organic agriculture, and third one is your genetically engineered crop based agriculture. Genetically engineered crop based agriculture. Okay, 3 type of agriculture can be performed. You'll be like studying about this agrochemical agriculture, organic agriculture, and genetically engineered agriculture के बारे में separately detail में पढ़ोगे in crop production and management. There is a separate chapter for you. Okay, this agrochemical agriculture means in this agriculture chemicals are used, chemical pesticides, insecticides, and other fertilizers are used. Chemicals are used in this agriculture. Organic agriculture means whenever you are using organic farming. Organic farming का तो नाम ही सुना है. Organic fertilizer का यूज करते हैं, different living organisms का यूज करते हैं. We use different living organisms as insecticides. We can use them as insecticides. Like dragonflies, they will eat the mosquitoes larva. Mosquito का जो larva होता है उनको खा जाते हैं, खत्म कर देते हैं. Along with that, other organisms are there like our earthworm. एक earthworm है अपना organism जो कि it will increase soil fertility. Whenever you are getting much amount of earthworm in any soil, if you are seeing much amount of earthworm in any soil, it means that oil is more fertile. Earthworm presence makes the soil more fertile. Okay, is this clear to everyone? And last one is your genetically engineered crop based agriculture. Means what? Nowadays many type of hybrid crop are produced. All these hybrid crops are genetically engineered. It means like their gene is changed. We have altered the gene, we have modified the organisms. That modification of the organism like modification of the gene of the plant, bacteria, fungi, animal, or any other organisms are called genetically modified organism. Is this clear to everyone? What are genetically modified organism? If you are altering gene of any organism, if you are changing or altering gene of the any organism, उसको कहा जाएगा तुम्हारा genetically modified organism. Is this clear to everyone? Genetically modified organisms are the organism whose gene is manipulated or altered. अल्टर किया गया है जीन को या फिर मैनिपुलेट किया गया है जीन को, उस ऑर्गेनिज्म को कहा जाएगा जेनेटिकली मॉडिफाइड ऑर्गेनिज्म दैट आर यूज्ड, या फोर्सफुली उसको अल्टर किया जाएगा शिवराज, एग्जैक्टली. Let us start talking about, let us take few examples of genetically modified organism. There are certain advantages of genetically modified organism and disadvantages also. We cannot modify human genome. Human genome modification is completely restricted. Human genome का जो modification होता है human के केस में, human genome का modification वो completely restrict कर दिया गया है इंडिया में, वर्ल्ड में और इंडिया में खास करके. Why is this restriction happening? Because human genome cannot be altered. Like it can be altered, but if you are altering this, this is completely illegal. Because people are using wrongly. Human genome को modify करके wrongly use किया जाता है. Many viruses, many bacteria निकाला जा सकता है जो कि ह्यूमन को अफेक्ट कर सकते हैं, इसीलिए ह्यूमन जीनोम को अल्टर नहीं किया जाता है. ठीक है, ह्यूमन जीनोम को मॉडिफाई नहीं किया जाता है. ठीक है, इलीगल होता है. यस, एडवांटेजेस ये है कि लाइक वी कैन प्रोड्यूस मोर प्रोडक्टिव and nutritive products. That's why this is an advantage of the genetically modification or genetically modified organisms. Okay, along with that we can study about certain diseases. Genetically modified organism को लेकर के we can study about diseases, different types of diseases. We can check vaccine safety. वैक्सीन सेफ्टी चेक कर सकते हैं, मेडिसिन सेफ्टी चेक कर सकते हैं. डिफरेंट इन डिफरेंट वे वी कैन यूज दिस जेनेटिकली मॉडिफाइड ऑर्गेनिज्म. विल बी टॉकिंग अबाउट दिस वैक्सीन सेफ्टी क्या होता है, स्टडी ऑफ डिजीज क्या होता है, एवरीथिंग को डिटेल्स में देखेंगे. ओके, लेट्स मूव टू द नेक्स्ट पार्ट. विल स्टार्ट विद द फर्स्ट एग्जांपल, दैट इज योर मेनी टाइप ऑफ पेस्ट रेजिस्टेंस प्लांट्स आर प्रोड्यूस्ड और इंसेक्ट रेजिस्टेंस प्लांट्स आर प्रोड्यूस्ड फॉर सच एज बायोरिएक्टर, बीटी कॉटन, BT corn, rice, tomato, potato, soybean. Different types of plant has been produced. Different types of plants has been produced. Okay, insect or bio pest resistance or insect resistance plant has been produced through this biotechnology process. Okay guys, आया समझ में सबको? हाँ, TB vaccine found in Garavis BT. BT means I'll explain you, don't worry. I got it out here guys, just wait. Sivraj, test tube baby. Test tube baby, we can say that it is not exactly biotechnology because we are not like altering the gene. इसको biotechnology नहीं बोल सकते. This is only technology used in biology. Okay, not exactly biotechnology. Anyway, we'll talk about BT cotton. BT cotton is an example of genetically modified crop. It is an example of pest mean insect. हर्ष पेस्ट का मतलब होता है इंसेक्ट. पेस्ट का मतलब होता है इंसेक्ट्स. ओके, जो कि क्रॉप को अफेक्ट करते हैं. बीटी कॉटन्स आर मींस दीज आर बीटी कॉटन्स आर जेनेटिकली मॉडिफाइड क्रॉप और प्लांट्स. व्हिच क्रॉप? दैट इज योर कॉटन क्रॉप. Cotton crop is a genetically modified crop. Here BT stand for Bacillus thuringiensis. BC का मतलब यहाँ पे होता है Bacillus thuringiensis. Okay, this is, listen this one carefully. Bacillus thuringiensis, listen this one carefully. Bacillus thuringiensis means this is a bacteria. What is this? This is a bacteria, right? This Bacillus thuringiensis bacteria के केस में क्या होता है, just wait a second guys, बताता हूं तुम लोगों को इन डिटेल. Bacillus thuringiensis का डिटेल बताऊंगा तुम लोगों को, ठीक है. What happened? Bacillus thuringiensis, they produces they produces this Bacillus thuringiensis has a gene, that gene is called as cryazine. This Bacillus thuringiensis has a gene, इनके पास एक gene होता है, these are bacteria, they have a gene, that gene is called as cryazine. This cryazine undergoes transcription, and it will produce mRNA, and after mRNA production it will undergoes translation. This is the normal process. Listen this one carefully. Bacillus thuringiensis is a bacteria that has cryazine, and that cryazine undergoes transcription and translation, and it produces a protein. That protein is called cryoprotein. That protein is called cryoprotein. After the class, read out the NCERT, and in NCERT you will get about cry-A and cry-B protein, cry-A and cryazine protein. तो जो होता है, first one is your cry-A. One second. Cry-A and cry-B. Go through this one. इसको समझ आ जाएगा अच्छे से, ठीक है? So these two genes will be there. They will be producing cryoprotein. Now the role of this cryoprotein is to, ये cryoprotein produce करते हैं, okay? But this cryoprotein is activated. This is inactivated. Normally ये inactivate होकर के रहता है. They will get activated. They will get activated only in presence of the saliva, or I can say only in presence of the alkaline medium. जब alkaline medium होगा तो at that time it will get activated. This happens in normal situation. Bacillus thuringiensis के केस में, okay, they have a cryazine, they undergo transcription and translation, and they produce cryoprotein, which is inactive, right? They will get activated. This protein will get activated in alkaline medium, and when they are getting activated, this will turn into a toxic substance. This will turn into a toxic substance. कैसा बन जाएगा ये? टॉक्सिक बन जाएगा, पॉइजनस बन जाएगा, टॉक्सिक और पॉइजनस बन जाएगा, टॉक्सिक और पॉइजनस ये बन जाएगा, राइट? दिस विल कन्वर्ट इनटू टॉक्सिक और पॉइजनस सब्सटेंस. आया समझ में? ठीक है. नाउ आई विल बी टेलिंग यू दिस वन. क्या होगा उसी Bacillus thuringiensis से? व्हाट वी विल डू, वी विल एक्सट्रैक्ट दैट बीटी जीन और क्रायोजीन को हम लोग क्या करेंगे? बैक्टीरिया से क्रायोजीन को विल एक्सट्रैक्ट दिस क्रायोजीन, एंड वी विल इंसर्ट दिस पर्टिकुलर क्रायोजीन. इसको हम लोग क्या करेंगे? इंसर्ट करेंगे. वी विल इंसर्ट दिस क्रायोजीन इनसाइड tobacco plant or cotton plant. We will insert it into the cotton plant. Now this cryogen inside the cotton plant will undergo transcription and translation. It will undergo transcription and translation, and it will produce that cryoprotein or crystalline protein or cryoprotein. ये synthesize करेगा. And I told you in the beginning only, these cryoproteins are inactive. ये inactive होते हैं, and when they are inactive, they are non-toxic. ये non-toxic होते हैं. Okay. Now what happens? Caterpillar का larva. Okay. Larva of caterpillar will come and infect this cotton plant. They will start eating over the cotton plant. They will start infecting the cotton plant. Initially, when the cotton plant is infected by this particular caterpillar ka larva of caterpillar, when it was infected by larva of pest, larva of caterpillar से जब ये इन्फेक्ट हो रहा था, तो इनिशियली आराम से ये वो जो कॉटन हो रहा था, कॉटन को खा के जा रहे थे. बट व्हाट हैपेंड नाउ आफ्टर दिस पर्टिकुलर रोल? ये होने के बाद अभी क्या हो जाएगा? नाउ व्हेनेवर दिस पर्टिकुलर लार्वा इज कमिंग एंड ईटिंग द कॉटन प्लांट, जब ये कॉटन प्लांट के ऊपर फीड करेगा, व्हेनेवर दिस लार्वा कम एंड फीड अपॉन द कॉटन प्लांट, it will swallow or it will engulf cotton plant. Along with cotton plant, it also, ये larva जो है, it engulf cotton plant. Along with cotton, it will also engulf this cryoprotein, crystalline protein और cryoprotein को भी ये engulf कर लेगा. Now this cryoprotein, they were inactivated, they were inactive in the plant. But when the larva is taking this one, when the larva is feeding upon the cotton plant is also like the larva is engulfing the cotton plant. Along with that cotton plant, cryoprotein is also going inside of the larvae. Larvae के अंदर जाता है. And in the larvae, what happens? Larva have some saliva. Larva के पास क्या present होगा? This larva have some saliva. Larva का क्या रहेगा? Larva का saliva रहेगा. And everybody knows that I told you in the early like beginning only. जो saliva larva का present रहेगा, that saliva will be alkaline. That saliva will be alkaline. अगर saliva alkaline है, it means this crystallite protein will get activated now. And if it is getting activated, it means it is toxic. तो ये toxic में turn हो जाएगा. This will turn into toxic substance. This will turn into toxic substance. Is this clear to everyone, guys? आया समझ में सबको? Initially larva feed कर रहा था. Initially larva was feeding over the cotton plant. Nothing was happening to the larva. Later on, what happened? When larva started feeding on, Nandis, I'll come to your doubt. Just wait. Okay. This larva will feed upon the cotton plant, and the cotton plant is now not normal cotton plant. This cotton plant is now BT cotton plant. अभी ये cotton plant का नाम क्या है? BT cotton plant. Because that BT gene or cryogene is present in this cotton plant. Larva is feeding upon the cotton plant now. It is swallowing or engulfing the cotton plant's leaves. Along with that leaves, inside the leaves, cryoproteins are also accumulating because cotton plant was producing the cryoprotein that was accumulating inside the leaves of the cotton plant. Right? So this cryoprotein will go into the saliva of the larvae of this particular caterpillar. Particular caterpillar के saliva में चला जाएगा. Caterpillar के saliva में चला जाएगा. Okay. And it will get activated in the alkaline saliva medium of the larvae. And whenever it is getting activated, it is turning toxic. ये toxic हो जाएगा, and it will, like this toxic substance will go and bind to the midgut of the insects. जो midgut होगा, midgut का cells में ये जाकर के epithelial cell में, midgut का epithelial cell में bind करेगा, epithelial cell में bind करेगा. It will bind to the epithelial cell of the midgut of this particular insect. Now what will happen? After binding, it will start perforation. क्या कर देगा ये? छोटे छोटे pore produce कर देगा. It will form some pores. Pores produce करेगा, छोटे छोटे pore produce करेगा in the epithelial cells of midgut of the insects. Insects के midgut में क्या हो जाएगा? Pore produce होगा. Epithelial cell में pore produce होगा. Now this pore formation leads to the destruction of this larvae, and larva automatically die. Larva का death हो जाएगा फिर. ठीक है? This is how BT cotton plant अपने आप को बचा लेगा. एक बार खाया, थोड़ा सा feed up किया. After feeding a little much only, it will die automatically after some time. So this larva will not develop into adult one, and it will not feed upon the cotton plant for the second time. If it is eating once, then it will get die. Okay, समझ आया सबको? This is how BT cotton plant is prepared so that it can prevent itself without any fertilizer, without any insecticide, and without any pesticide. This BT cotton plant can prevent itself from the infection or protect itself from other insects. Is this clear to everyone now? I hope this is clear to everyone, guys. ठीक है? Now I am coming to your doubts पास. बहुत सारा doubts I got here. ठीक है? मैं बताऊंगा तुम्हारा जो भी doubts है मैं बता दूंगा. ठीक है? From where it is extracted? Sinchana, this is extracted from Bacillus thuringiensis bacteria. Bacteria is a Bacillus thuringiensis is a bacteria. From there only this cryogene or crystalline gene is extracted. ठीक है? None these crystals means small particles, minute particles जिसका like rough surface होगा. जैसे सुगर का होता है, सुगर सर क्रिस्टल, सल्ट सर क्रिस्टल. ठीक है? ठीक है हर्षा, मैं शॉर्टली रिपीट कर दूंगा, नो प्रॉब्लम. क्रायोजेन का फंक्शन मैंने बोला, दे विल बी सिंथेसाइजिंग क्रायोप्रोटीन, एंड क्रायोप्रोटीन लेटर ऑन स्टेजेस में टॉक्सिक हो जाएगा. दैट विल बी कन्वर्टिंग इनटू टॉक्सिक सब्सटेंस, एंड इट विल like kill the insect or pest. ठीक है, पेस्ट मीन्स बताया था मैंने हर्षा, व्हाट इज रीजन इफ दे आर टॉक्सिक. They are toxic, like what is the region of it? They are toxic. It's not like any region. Okay, few things are toxic because of presence of like chemicals, but here we need not to go in depth of that thing कि क्यों ये इतना toxic होता है. Just remember this one, ये क्रायो प्रोटीन जो होते हैं, जो क्रिस्टलाइन प्रोटीन्स होते हैं, they are toxic. ठीक है शिवराज, ठीक है. Plasmid of gene or cotton by the gene. If the rain is alkaline, it will be toxic for everyone. If the rain is alkaline, it will be toxic for plant also. But remember this one कि plant के केस में जो होगा ना, in case of plant, the rain whichever like alkaline rain or acid rain is happening, that acid rain goes into the soil, and in the soil only it will get purified. Okay, very less amount of alkaline water will absorbed by the plant, and that will not affect anyhow. That will not affect to this plant. ठीक है, क्रायो प्रोटीन का एक्टिवेशन में वो हेल्प नहीं करेगा. ठीक है, आया समझ में? इट्स नॉट लाइक दैट, क्योंकि जो प्रोटीन है, दैट प्रोटीन, सी नंदिस का डाउट ये है कि कॉटन क्लॉथ वी यूज, देन if the acid rain is happening, then cotton cloth is also getting toxified or what? No, not exactly like that, because the protein, that is living thing. Okay, protein जो होता है वो बायोमोलिक्यूल्स होता है, but in cotton dresses or clothes it is not in activated form, so it will not get activated once again. Inactive भी नहीं है, activated भी नहीं है, proteins will be like in like dead form. ठीक है, हाँ, दैट्स ए वेरी गुड क्वेश्चन सिंचना का बहुत अच्छा क्वेश्चन आया है. सिंचना का बहुत अच्छा क्वेश्चन आया है. दैट क्वेश्चन इज लाइक no, this will not affect the cotton, Deepika, because in cotton plant it is not affecting the cotton plant, because see, this cryoprotein are inactivated in form, inactivated in the plant only. They are produced in inactivated form. They will be getting activated in presence of the saliva of this particular caterpillar or insect. ठीक है, सिंचना का डाउट ये है, सिंचना एज ए डाउट, सिंचना इज आस्किंग when this particular organism is dying, which organism? This caterpillar is dying, it will like fall in the soil only, and in the soil decomposers and all other earthworm and all other consumer or decomposer will be there. Will it not affect them? Remember this one, Sinchana, every toxic substance in the soil will be digested by decomposers. They are decomposers, it will not affect them, and decomposers are there to digest the toxic substance and all other substance. They will digest this toxic substance and they will convert it into the non toxic substance. Okay, and वही non toxic substance फिर अपने बॉडी में आता है. ठीक है, हाँ, this will affect all type of caterpillar, all type of insect which have saliva, which have alkaline saliva, okay, सारे caterpillar, सारे insects को ये affect करेगा. ठीक है, I took example of caterpillar only, but ये सारे caterpillar and insects को affect करेगा जिसका saliva alkaline है, including human if he eats. Exactly, our saliva is alkaline, then it will also affect our body, but it will not affect that much in our body. ठीक है, क्योंकि इसका epithelial cell को ये perforate करता है, but in our case it will not perforate our epithelial cell that much. Okay guys, any other doubts, please ask. Okay, please ask if you have any other doubts. Alright guys, I'm launching teacher NPS poll now. Please vote in the teacher NPS poll and keep asking your doubt. I'll be solving your doubts. ठीक है, cow के केस में probably it will be affecting, but Nandis, this way I can say like these cottons, cow के केस में ये जो बेसिलस thuringiensis होगा, ये इतना ज्यादा अफेक्ट नहीं करेगा, एपिथेलियल सेल को इतना ज्यादा परफोरेट नहीं कर पाएगा, बिकॉज आवर एपिथेलियल सेल इज समहाउ डिफरेंट फ्रॉम द एपिथेलियल सेल ऑफ कैटरपिलर. ठीक है, सो इट विल अफेक्ट द इंसेक्ट्स का एपिथेलियल सेल को, राइट? ओके, 3 and this is running 4th year, Madhu, 4th year running. ठीक है, 4th ईयर रनिंग। ओके, हर्षा का कुछ डाउट है, सेप्टिसेमिया मींस, सेप्टिसेमिया, ओके, सेप्टिसेमिया मींस लाइक इसका इसका जो which one, like epithelial cell, it will get like, it will get septate, or septate I can say, it will get divided or fragmented into many pieces, and that is for your septicemia larva, dead larva. Okay, why does this toxin not kill the bacillus? Yeah, because this toxin is not in activated form in bacillus. Bacillus thuringiensis बैक्टीरिया के केस में ये जो सिंथेसाइज होता है क्रायोप्रोटीन, दैट इज नॉट इन एक्टिवेटेड फॉर्म. Okay, that remain in the bacteria in inactivated form. That's why. Madhu, me and Subramanya, both of us have the same, like we joined at the same time only. Okay, ठीक है. नॉर्मल कॉटन, हाउ दे आर मेड इनटू बीटी कॉटन. ओके, नॉर्मल कॉटन में जो भी प्रोसेस आई एक्सप्लेन यू इन द बायोटेक्नोलॉजी प्रिंसिपल एंड प्रोसेस, प्लाज्मिड एंड ऑल अदर थिंग्स, जीन ऑफ इंटरेस्ट विल बी इंसर्टेड इनटू द कॉटन, एंड नाउ okay, what I told, okay, well, normal bt cotton cell will be there, normal bt cotton will be there, that bt cotton will be inserted with gene of interest. That cotton will be inserted with gene of interest, which gene, cryogene, will be inserted into bt cotton plant by the help of plasmid or any other things. Plasmid की मदद से या फिर any other things की मदद से कर सकते हैं. ठीक है, ओके. हाँ, जीन गन मदद से, जीन गन, दिस इज प्लांट, सो वी कैन अप्लाई लाइक जीन गन, वी कैन यूज जीन गन. Not exactly, not exactly, Sinchana. Because for the activation of this cryoprotein, what we need some alkaline medium, and in bacteria alkaline medium is not maintained, that's why it is inactivated in bacteria. ठीक है, ठीक है. I did MSc, Madhu. Okay, guys. Okay. Thank you so much. Thank you so much, guys. Any other doubts? Please keep asking. ठीक है. So keep asking if you have any doubt. 1 second, huh?